What's up, biology students? Mr. Holloway here, but you already knew that, of course. In today's video, we're going to talk about passive cell transport, one of the means by which cells maintain homeostasis and remain in balance with the extracellular environment. But before we start talking about cells, I wanted to tell you a little story about fish. When I was younger, I had an aquarium where I kept freshwater fish like goldfish and guppies and mollies and the like. Now, if you've ever been to a pet store and walked into the fish section, you've probably noticed that they've got both a freshwater fish section and a saltwater fish section. You might also notice that many of the saltwater fish are like way prettier than the freshwater fish, like the beautiful, brightly colored saltwater fish we see here on this slide. Well, one day, I'm at the pet store buying a new fish for my aquarium, and I overheard this guy talking to the store attendant asking about the saltwater fish. The attendant was explaining to him that no, you can't just buy one of these saltwater fish and put it into a freshwater tank because it will die if you do. Well, won't the fish just evolve? The guy asked. First of all, that's not how evolution works. But if you ignore that for a second, there's actually kind of an interesting question there. Why can't you put a saltwater fish into freshwater tanks? Understanding the answer to this question will also help you to understand why you can't drink seawater, even if you're stranded in the middle of the ocean with nothing else to drink. The reason you can't drink seawater, and the reason you can't take a fish out of the sea and drop it into a tank of fresh water, has everything to do with cellular transport, specifically passive cellular transport, which is the movement of materials into and out of the cell. Because this happens due to the random motion of particles, cells do not need to expend any energy in order for this kind of transport to occur, which is why we call it passive cellular transport. Cellular transport is regulated by this remarkable structure called the cell membrane. Every living cell is surrounded by a membrane made of a phospholipid bilayer, a double layer of these phospholipids with this hydrophobic or water-repelling layer in the middle that keeps the cell's insides contained while controlling what can get in and out of the cell. We refer to this membrane as being selectively permeable or semi-permeable because only certain substances can pass through it. In this way, the cell membrane regulates the cell's internal environment and plays an important role in maintaining homeostasis. Although some forms of cell transport do require energy in order to move substances across the membrane, many important substances can be moved into and out of our cells via passive cell transport, which requires no extra input of energy in order to occur. Passive cell transport occurs by way of diffusion and osmosis, which you may have heard of before. Passive transport happens because of a concentration gradient, which means that there is a higher concentration of solute or dissolved particles on one side of the membrane than there is on the other. Think of a concentration gradient like a hill. There's a high concentration at the top of the hill and a low concentration at the bottom. A ball will naturally roll downhill, and particles will naturally roll down a concentration gradient from high to low concentration without you needing to help in any way. And again, that's why we call all of this passive cell transport. In this picture here, we would say that the outside of the cell is hypertonic, meaning it has a higher concentration of solute, and that the inside of the cell is hypotonic, meaning it has a lower concentration of solute. All of these solute particles inside and outside of this cell are moving around all of the time. And in fact, particles are crossing this membrane in both directions. But because there are more of these particles on the outside of the cell than on the inside, that means that there will be more of these particles colliding with the cell membrane, meaning more chances to move through it. With more of these particles on the outside moving into the cell than particles on the inside moving out of it, the concentration of solute inside the cell will gradually increase, while the concentration of solute outside of the cell will gradually decrease. And the higher the difference in concentration between the inside and the outside, the faster this process will happen. This process continues until the concentration of solute on each side of the membrane is roughly equal, at which point we would say that each side is isotonic compared to the other. Now our cell is in equilibrium, meaning that the rate of particles moving into the cell is equal to the rate of particles moving out, so the concentration remains balanced on each side. We call this natural movement of particles from an area of high concentration to an area of low concentration diffusion. Oxygen and carbon dioxide are both transported into and out of our cells in this manner. In the case of oxygen and carbon dioxide, these small, non-polar molecules are able to pass directly through the cell membrane. But larger molecules, like glucose, as well as polar molecules like water, have a very difficult time passing through the cell membrane directly. Transport proteins, or protein channels, embedded in our cell membrane help to move these kinds of substances into and out of our cells. We call this kind of diffusion facilitated diffusion because the process is facilitated or made possible by the presence of transport proteins. In most cases, the shape of the protein determines the kind of molecules that can be transported through it, and if that molecule has the wrong shape, it will not be able to fit through the protein channel. Let's see a moving example of how this whole facilitated diffusion thing works. Let's suppose that the top half of this simulation represents the outside of the cell, and the bottom half of this simulation represents the cytoplasm that fills the inside of the cell. In this simulation, we can see that there is a very high concentration of this green particle outside of the cell, and a very low concentration of this particle on the inside of the cell. We would therefore say that the outside of the cell is hypertonic compared to the cytoplasm, or we could say that the cytoplasm of our cell is hypotonic compared to the outside of the cell. Right now, these solute particles have no way to pass through the membrane because there are no protein channels of the right shape to facilitate the diffusion of these particles. However, if we place some protein channels into our simulation, we should see that diffusion begins to occur almost immediately. If you watch closely, you should notice that green solute particles are passing through these channels in both directions, but more of them are being transported into our cell than are being transported out. So the concentration of green particles in the cytoplasm increases, while the concentration of green particles outside of the cell decreases, until eventually both sides have a roughly equal concentration of solute, at which point we would say that we have reached equilibrium, and our cytoplasm is now isotonic compared to the outside of the cell. Now let's add a different solute to the cytoplasm of our cell. This blue particle has no protein channels to pass through, so its concentration inside the cell remains high. But as soon as we put some appropriate protein channels into our simulation, these blue particles begin to diffuse out of our cell, and the concentration on both sides of the membrane starts to equalize. If we let this simulation run for a while, we will see that both green and blue particles are still moving back and forth across the membrane in both directions. But the movement of particles into the cell is equal to the movement of particles out of the cell, so the overall concentration of particles doesn't change very much once we reach equilibrium. And that is the essence of homeostasis in this case. Water is an important example of facilitated diffusion because it is quite difficult for water to pass through the nonpolar, hydrophobic lipid bilayer that surrounds the cell. We call the movement of water through the cell membrane osmosis, and this happens by way of special transport proteins called aquaporins, which have just the right shape to allow water molecules to pass through the cell membrane. So far, so good. Stuff moves from areas of high concentration to areas of low concentration, and we call this natural movement diffusion. We call a solution hypotonic when it has a lower concentration of solute than our cell, and hypertonic when it has a higher concentration of solute than our cell. When the concentration of solute is equal on both sides of the membrane, we would say that our solution is isotonic. But it gets a little bit more complicated than that. Suppose that the green solute particle in our figure here cannot pass through the cell membrane, but water can. Because this membrane contains aquaporins, that allows osmosis to occur. Some of the same basic principles apply, but in this case it will be the water that diffuses from an area of high concentration to an area of low concentration. In this first figure, our cell is placed into a hypotonic solution, so there is a lower concentration of this green solute outside of the cell. But since the green particle cannot pass through the membrane of the cell, the only way to balance out the concentration is for water to diffuse into the cell. Think of this like having a higher concentration of water outside and a lower concentration of water inside. So water diffuses from high to low, from outside of the cell to inside of the cell. Likewise, if we place our cell into a hypertonic solution with a higher concentration of solute outside of the cell and a lower concentration of solute inside the cell, then the water will diffuse out of the cell to balance out the concentration of solute because the solute itself cannot pass through the membrane into the cell. This kind of thing happens in our bodies in regards to salt, and water moves into and out of our cells via osmosis, keeping the concentrations of salt and water balanced in and out of our cells. We call this osmoregulation. In these pictures, these small purple particles represent the salt. Since salt cannot pass through the membranes of our cell via diffusion, osmosis causes the net movement of water into the cell in our first example and out of our cell in the second example. In both cases, we can see that in the end, the concentration of salt is balanced both in and out of the cell, but that occurred because of osmosis, not because of the diffusion of the salt itself. This is why eating a lot of salty food makes you thirsty. When you eat all that salty food, the concentration of salt in your blood increases, much like in our second example here. With a higher concentration of salt in your blood than in our cells, water diffuses out of your cells in order to balance out this high concentration of salt in your blood. That makes us feel thirsty because our cells are losing water. This is also why it is unsafe to drink seawater, which has a much higher concentration of salts than our bodies have naturally. Drinking seawater would actually cause your body to lose water overall and would only make you more thirsty and more dehydrated in the long run. The balance of salt and water is important for all living organisms, and when cells are in an environment where the concentration of salt is very different than the concentration of salt inside their cells, then bad things can happen. In extreme cases, cells placed in a hypotonic solution may swell or even burst from the excess of water diffusing into that cell. Likewise, cells may shrivel up when placed into a hypertonic solution as water is lost through osmosis. Remember our question about fish at the beginning? Well, these very same principles apply to that situation too. Fish live in the ocean and in freshwater, but very few fish are able to live in both places. Saltwater fish are able to live in saltwater because their cells are in balance with the surrounding environment, and the cells in our saltwater fish are just as salty as the oceans in which they live. Put that ocean fish into a freshwater environment where the concentration of salt is much lower, and osmosis will cause water to diffuse into that fish's cells to balance out the salt concentration. This throws off the internal chemistry of our fish, who is accustomed to living in saltwater, and that fish will die as a result because it cannot maintain homeostasis in this new environment. Likewise, if we put a freshwater fish into the ocean, the fish's cells will suddenly be surrounded by an environment that is much saltier, and water will diffuse out of our fish's cells to balance the concentration of the environment around it, causing our fish to dehydrate and die. This poor freshwater fish will not be able to maintain homeostasis in a saltwater environment. In summary, diffusion and osmosis occur naturally and without cells needing to expend any energy to make those processes happen, which is why we refer to these forms of cellular transport as passive transport. While these processes help our cells to take in certain nutrients and materials like oxygen and glucose, and to expel waste products like carbon dioxide, these same processes can also cause problems if they cause our natural salt and water balance to get thrown off from what we're used to. For us to maintain homeostasis, maintaining a proper salt and water balance is key. So don't drink seawater, and if you eat a lot of salty food, remember to drink a lot of water to go with it. And with that, I will bring our video to a close. Thanks for watching, everyone.